Good afternoon to all. Welcome to the afternoon session of day 8 of FDP. So today we have our guest speaker, Dr. Farah Deba. She is an associate professor at CMR University, Bangalore. Prior to this, she has worked with Monsanto company as a senior scientist. And she holds a PhD degree in agricultural entomology and plant pathology from the GB Pant University of Agriculture and Technology, Pantnagar. She has further specialized in intellectual property protection from Indian Institute of Science, Bangalore in the year 2005. She has more than 20 years of experience in the field of intellectual property, agriculture biotechnology, R&D, teaching, research guidance, research communication and scientific outreach. Her areas of work include IP protection, agriculture biotechnology, competitive intelligence, technology prospecting, knowledge management, and data mining. She has several publications in peer-reviewed journals, including those of BT Cotton, BT Corn, and IPRs. She is an inventor with patents in genomics. Apart from her scientific publication, she also has literary work to her credit. These includes 2 English novels named Corporate Curry and Rain Shadow, and other 3 books on various topics. She is an accomplished vocalist specializing in ghazal singing and has a music album to her credit. On behalf of the Oxford College of Engineering, Department of Biotechnology, I, Salma, take it a privilege to extend a very warm welcome to Dr. Farah for being our speaker for this session. We welcome you, ma'am, and it is over to you to start the session.
Thank you, and good afternoon, everybody. So I'll share my screen now, and please let me know if I'm clear.
Yes, ma'am.
Okay, is it audible and is it visible?
Yes, ma'am.
Okay, then let's start. What we are trying to do at this stage is, I'm 100% sure that you have covered a lot of ground before this. And what I'll try to do is provide a bird's eye view of several things, and we'll go in a sequence. So why is it important to understand the whole process and how to go about the whole?
Excuse me, ma'am.
Yes.
If you could minimize your Teams icon.
Okay. Minimized. One second. Is it okay now? Okay.
Yes, ma'am, it is fine now.
Okay. So let's go through this whole discovery and commercialization pipeline and understand where we have to capture the IP. So in any industry, there is a sequence of things from a startup or from the beginning of the idea till the commercialization of the idea, there are several aspects. One is technical, another is legal, and then business. All these three have to come together in a form that you can take your idea or your invention to a logical level, which is to commercialize it for the benefit of the society. So from a provisional patent to an application to a patent to generating a whole portfolio of patents, it's very, very important to understand where and how, what due diligence is required. So when we talk about any commercial aspect, we have to be consciously doing our lab work, at the same time capturing the IP, and then you go ahead and develop a product, which is called product development. So from an idea which you feel that has a potential, and all the documents are required in the lab records and in the invention disclosure when you go to your attorney, then if it is worth, you'll go and file a provisional patent to get a priority date, and simultaneously you will be doing your other details, more specific testing, and then you go to the pilot testing. Meanwhile, parallelly, the patent office actions will happen, and you'll have your patent application done, and then 48 months will go. In the meanwhile, you do the pilot testing, you go to the venture capitalists and get your fund, and so on. And this process continues. Even after you launch one product, you will have to develop a portfolio of products and a portfolio of patent applications and patents around that, so that you capture your IP in a clear manner, so that you don't have to deal with the competition, or you have to effectively deal with the competition. Why is it important? As we go through, we'll understand. So let me give you a simple background how the biotech R&D landscape looks like. So it all started in 1980s, Diamond versus Chakrabarti. This has been heard several times, that originally his patent application was rejected because he was trying to patent a bacteria which was useful for breaking down crude oil. Then later on, they had to go to the Supreme Court, and ultimately they said anything under the sun that is made by man is patentable because there is a human intervention. So it's a live human made microorganism. It's a composition of matter. So this happened in 1980s. But now the definitions and scope, we have learned a lot in this period from 1980s to now. All these definitions, the scope, the inventions and innovations that have happened have evolved dynamically. We now have better guidelines because the patent offices also understand the subject better now. And we have case laws. So when a case goes to the court will deliberate on it and create laws which are called the case laws, and they become the precedent to follow. So in the same time, when this biotech invention was given a patent, in US there was a Bayh-Dole Act, which allowed the universities, the academic institutions, to generate patents, and then they started creating offices of technology transfer, incubators, and startups. So this parallelly happened, and it allowed the industry to grow. So this is very important to understand because the core enabling biotech research happened in the academic institutions, mostly the enabling technologies. And this work continues in the academic atmosphere, the discovery and functional genomics, etc., in an exploratory manner. But meanwhile, the industry jumped in and they started using the patenting and creating products. So now there are differences between what the public sector does, which is the academics, and what the private sector does, which are the companies. So here we go for the knowledge-based things, while the commercial objectives are different from the knowledge-seeking activity. So that's the difference. And when we see all US patents, the public sector holds very few patents, while the private sector has a lot of patents because they go to the market. But when you see this unique area, which is the biotechnology, you'll see that the public sector holds a huge chunk. So it's a unique position for biotechnology. This data is for ag biotech, but this is as well applicable for all kind of biotech. Why is it important to understand this aspect? Because it's a multi-component technology. You need many things. And the genes that are conferring a trait, then what are the pathways involved? What are the tissue culture technologies, the cell lines, and so many other enabling technologies are required. So it's a multi-component technology, and you need to understand various aspects. And bits and pieces of this work happens in different labs. So how do you bring all these together to create a product? So this R&D landscape is highly technology-driven, knowledge-driven, and highly competitive because it's a multi-component, complex technology. The research is capital-intensive. It requires long-term investment, and also the need of key enabling technology, the basic technologies which are required to generate a genetically modified organism. And in case of agriculture biotech, they also need the crop. They need to own the germplasm or the crop variety. And there are different agroecological zones. Everything doesn't work everywhere because it's a complex ecosystem. So these are the prerequisites. Now, the fundamental research happened in the academic institutions, but then the private sector started using stronger instruments for IP protection. So this whole scenario of R&D in biotech became a very interesting subject to study. And as a result of this fast emerging breakthroughs, there was ambiguous and complicated freedom to operate. This word freedom to operate is very, very important to understand, because when we talk about patent rights, we also talk about infringement. We also talk about defending our whatever work we have done. So it's very important to understand what is freedom to operate. In a new age, knowledge intensive situation, where every year we see breakthroughs and powerful research tools without which this sector cannot work. It led to ambitious product pipelines and fierce market competition. And it's a kind of a revolution. Initially, pharma and agri-biotech both were doing very well, but agri has gone down now and pharma stays strong. And we see these interdisciplinary technologies, which are nanotechnologies, bioinformatics, systems biology, computational biology, and AI. And the third area right now looks like the brain, the antibiotics, which we have to reinvent, battling cancer, the gene editing technique, we'll talk further on that, organ generation, and stem cells. So these are the thirst areas. And we broadly categorize it as agriculture and food biotech, industrial biotech, so on and so forth. So what happens? When you try to develop a drug or a agri-biotech product, you go through a pipeline. So you will generate data. In the first phase, there will be a data generated, and then some of the material will go to the next phase, and some will be rejected in the pipeline. And this will go on for several years, in fact. So you will check the efficacy of a drug, we'll check further in different populations of the patients, and further on in diverse populations. So what happens is that as the data is generated, you have to capture the IP at each stage. And as you go through the office actions in the patent office, you will progressively define your scope and claims better. And this is highly capital intensive. You see the 9% is the cost in lead discovery. But once you develop the lead, you'll go to the optimization stage, preclinical, phase 1, phase 2, and so on. By the time you come to phase 3 and submission, a huge chunk of spent. So when you see this situation where there's high investment and a long drawn process, so due diligence is required right from the beginning, from the drug discovery. You have 10,000 to 5,000 compounds. From that, you'll go to the preclinical where 250 compounds will be there. Then clinical trial may happen with 5 or 6 compounds. Ultimately, after several years, you will get one FDA-approved drug. And then you go to the market and face the market competition. So these are the steps where at each stage, we have to clearly capture the IP and audit what is not important, leave it behind because the resources are limited, and take forward potential candidates which may give you a product. And then as we progress through the research, we'll write more precise claim that this is important for this disease, these are the other applications, and so on. Now we see the examples of some active pharma molecules. So in the first generation, we have the ibuprofen, then we have this peptide, which is for diabetes, then we have a protein for breast cancer, and further on double-stranded siRNA for treatment of genetic diseases. So these are the kind of molecules that we are coming up with now. So the earlier generation had chemical molecules. Now we have peptides and proteins and siRNAs, and so on. So this is very important to understand that from a simple form, we are coming to more complex forms and more specific diseases that we are trying to cater to. And we don't work in isolation. We have to, when we draw our timelines, we have to understand what is the regulatory framework, from where we have to get our approvals. So we need to understand that. So we have the WTO TRIPS, we have the United Nations, the World Health Organization, and so on and so forth. So regulatory compliance throughout the phase is very important. So for Ag Biotech, USDA, APHIS, you have to take permits from them if you want to do trials in the field, from Food and Drug Administration for all the drugs, Environmental Protection Agency for all the pesticide trials. So these are the regulatory bodies that you have to deal with in US. Similarly, we have protocols. How will you handle, how will you transport the living modified organisms? And there's a protocol, Nagoya Protocol, which says whether you can access the genetic resource and equitable sharing of benefits, which should be fair. In India, we have the Drugs and Cosmetics Act. So the Drug Controller General of India and the state bodies will control this. Then for all the Ag Biotech and other things also, we had to go through GEAC and RCGM, and institutional biosafety committees were instituted. And there are recombinant DNA safety guidelines. So all this infrastructure is there and we have to go through them. So we have to make sure that we keep this in mind, that we have to go through this regulatory process and keep our timelines accordingly. And these biotech products have developed very fast and they are again developing dynamically. So we can have patents, we can have plant breeding rights, we can have plant variety protection, and so on. So now, what happens is that everything is territorial. All the laws are territorial. What happens in one country does not happen in another country. So we need to understand in different geographies what is patentable and what is not patentable. So in India, inventions in atomic energy, so on and so forth, they say that you cannot patent these. And what is relevant to us is that morally objectionable scientific endeavors like human cloning, terminatal technology, oncomouse are not patentable in India. And Dolly, you know that it's a clone, so cloning is also not allowed in India. But when you see it in detail, initially there was a lot of confusion, and there was an amendment done which says the mere discovery of a scientific principle is not patentable. So then if you have discovered a microorganism which is an extremophile or something like that, can you patent it or not? Then it says treatment of human beings, similar treatment of animals to render them free of diseases. So what they did in this, they removed the word plants. So now it was, this deletion allowed one thing, that is the method of modification of plant, a genetically modified plant was allowed by deleting that or plants. So you see how the law is and how it applies to real life conditions. Then it says plants and animals in whole or any part thereof are not patentable other than microorganisms. That means you can patent microorganisms, but essentially biological processes cannot be patented. Okay. So you can have the process. So there are two kinds of patents: a product patent, say a drug molecule, and a process patent, the process of creating a product. So you can either have a product patent or a process patent. So in case of GM crops, we did get a process patent. So now what is patentable is transformed plants. This is not patentable. Essentially, biological process is not patentable, because if you have just a gene sequence without disclosing their function, there is no inventive step. So you cannot have patent. What is patentable is microorganisms, transgenic microorganisms, recombinant DNA, where you have shown substantial human intervention. So please remember that substantial human intervention. The process of developing a transgenic product. So as I said, it's not a product patent, but a process patent, how to develop a transgenic product. So that process is patentable. But there were still gray areas. For example, it is not defined what is a microorganism. And what about biological materials? So this I have just listed. This is my list, which should be in the legal form. It should be accepted what you can patent and what you can define as microorganisms. So now, when we go to the European Patent Office, they have come a lot further, and they said that genes and nucleic acid molecules, such as disease-related genes for diagnosis or antisense siRNA for therapy, proteins such as insulin, enzymes, proteases for washing powder, antibodies for cancer treatment. These are all patentable. Virus sequences for testing and therapies, cells such as stem cells, microorganisms for bioremediation, plants which are herbicide resistant, golden rice, we'll discuss further on golden rice, which is vitamin A and enriched rice, and animal disease model. So here comes the GM oncomouse, which is required for testing for cancer. So it's a model for testing cancer. So these things are patentable in European Patent Office. But sequences without known function, ESTs, are not patentable. Genetically modified animals which suffer but are not associated with medical benefit, substantial medical benefit, such as just testing and using an animal to test some cosmetics, is not morally good. So they will not accept it. So plant varieties, which are protected under UPO, which is the plant variety protection. And similarly, animal varieties, they are not patentable, but you can go to the UPO and protect them as a variety. Now, what is not patentable is human embryos or human germ cells. Human animal chimera is not allowed. Destruction of human embryos not allowed. So these are not patentable and not allowed. So again, the story is that in the 1990s, there was a huge patenting activity scene, and people were making jumbo patents with hundreds of gene sequences listed. They had some known functionality, some unknown functionality. And because there was a huge rush, there were a lot of business realignments, and there were joint ventures, and small companies were acquired, and mergers were happening. So this is one of the patents where I'm also an inventor. This is a Monsanto patent, which says transgenic plants with enhanced agronomic traits. So it's as broad level as possible. So you see that date of filing is 2007. Application came out in 2011. So what it does, it says there's a recombinant DNA, a promoter, an amino acid sequence, a PFAM domain, so on and so forth, and says enhanced trait. Then there it lists enhance water use efficiency, cold tolerance, heat tolerance, so on and so. So everything is exploratory, non-specific, and they're trying to claim everything. So everything will not be given actually, because this is too broad a claim. And this is just the first claim. And this is the amino acid sequence associated with that patent. So with all this, we evolved. And in 2013, there is a case which says AMP versus Myriad Genetics. So Myriad Genetics, they got a patent, 7 patents for breast cancer genes, BCRA1 and 2. And then they had claims which said the code of both the genes, references to locate those genes, and identifying the mutations in the patents. So these things were there. The USPTO practice was to allow usage of isolated DNA sequence as a composition of matter. So you have isolated something, you can patent it as a composition of matter. But this case overturned all that. So just isolating something. So there was a case where they allowed adrenaline to be isolated, and then they got a patent on it. This was 1911. But in 2013, the Supreme Court invalidated that practice in the US and said any naturally occurring DNA segment is a product of nature and not patent eligible. Merely because it is isolated, you cannot have a patent. But when you make a complementary DNA, that will take a process. That complementary DNA is patent eligible because it is not naturally occurring. So now you see the difference how we have evolved that concept. So isolation itself is no longer sufficient. You have to go through a process and then claim that we have gotten this after a process, which is a cDNA in this case. So this is a landmark case, and it was in 2013. So now you have created something, and you know what you can patent and not patent. But then how will you monetize it? You have spent so much of money, so we should know how to manage our IP. Because from the concept phase to taking it to a patentability analysis where we'll see whether to patent or not to patent, then to patent filing, then prosecution. If there's an application, is there an objection to the application? If there's a patent granted, is there an objection to the patent? And then maintaining it. So the term given is 20 years, but most of your time will go in going through this whole exercise. then by the time you reach those 20 years, much before that, another technology will come. So you have generation 1, 2, 3, 4, and now you have 5th generation. So every time we change our mobile phones, because there's a technology life cycle, and technologies will become obsolete over a period of time because parallelly several people are working in the same area. So that we should understand that other people will come up with something different. So when we see this 2018 European Patent Office report, it gives clearly medical biotechnology, medical technology, biotechnology, pharmaceuticals, all these are in the top 10 technology fields. So now when you have so much of patenting activity happen, what will happen? Confusion. So a dense web of overlapping IP. Okay, you have to hack your way through this if you want to commercialize a new technology. Because not only you, several other people are working in the same area. So it will impede commercialization. Because just like other complex technologies, biotechnology also is modular and highly complex. Different components and tools are required to create one product. So we have to understand where we are existing in which kind of a situation. So if there's a partial or a complete overlap of functionality, the patents will be overlapping and then there will be a confusion. So we call this a patent thicket. There is so much of confusion because in that technology area, there is a lot of activity happening. I'll give an example of golden rice where 70 patented technologies were used in R&D and 44 US patents are required for practicing this invention. From rice, they have added genes which allow more precursors of vitamin A. So the resultant rice is rich in carotenoids. Okay. So the ownership was diffused. There was a lot of litigation and uncertain freedom to operate. Nobody knows how to go about it because it is so complex and everybody is infringing everybody else. This is called reciprocal infringement. So what happened? They had to create a system where they secured the rights from different companies, put it in one organization, and then said, this technology license is only for humanitarian use, no commercial rights given. And then it went into a greenovation company and sub-licensed to the IRRA. And there also, there are ifs and buts and what kind of license. We have to understand the situation when we are working in this field. And then due to all this, from 1980s till now, there were a lot of wars, patent wars. And ultimately what happened, there were so much of infringement and counter infringement that these 6 organizations, the 6 big companies in life science, they ultimately merged together. You see the Monsanto has gone to Bayer, Dow, DuPont, and Pioneer have become Corteva. Syngenta has gone to Chem China, and we have BASF. So we have lesser and lesser players, which is a monopolistic situation. So when we take the example, the first is Monsanto legal cases. So Monsanto heavily invested in R&D, and then there were high-profile lawsuits which were happening. Initially, they aggressively patented, as I showed that jumbo patent, and then defended. So this is the agrobacterium, which is we all know is a natural genetic engineer, and the TI plasmid is required for the transformation. So this was a natural thing, and there was a huge case between Monsanto versus Max Planck Institute. And ultimately, Monsanto lost, and Max Planck won in European Patent Office as well as in USPTO, and the agrobacterium plasmid rights were given to Max Planck. So in case of Monsanto, they were doing the work in a dicot plants and the Bolgard insect protected cotton. I was working on this. This was developed using this agrobacterium mediated transformation. So these are the real cases what happened in the 1990s. Similarly, between Monsanto and DuPont for herbicide resistance genes, there was a case then about the Bacillus thuringiensis between Monsanto, Syngenta, Dow, and so on and so forth. So all these cases have happened, and because of the complexity of the technology itself. But later on, they ran out of ideas and they started cross-licensing within themselves and stacking the traits. 2 companies coming together and putting one herbicide tolerance gene, one another herbicide tolerance gene, and so on, and insect tolerance. And they created these. And later on, they ran out of further ideas. So now another very big example is the CRISPR-Cas9 system for genome editing, which is very much in news, still very much in news. And there were two labs working on it. One is the Jennifer Doudna's lab in Berkeley, UC Berkeley, and the other was Fang Zhang in Broad Institute. So now these two people were caught in this war. And you know that she recently got the Nobel Prize in Chemistry. So this is a genetic editing technique which is used for developing therapies and so on. Then we have also seen the examples of biopiracy where without approval, people have gone ahead and used somebody else's. So one of the examples is turmeric. They patented it for wound healing. And then India generated a whole controversy here. And then people went back and provided texts and showed that this was prior knowledge. So the patent was revoked on the basis of lack of novelty. Similarly, Rice Tech, they tried to create something which is like basmati, and they claimed for that rice plant based on starch index. So method of selecting based on starch index. So, but they were clearly trying to deceive the customer by saying that this is kalmati, texmati, and so on. So it's a collective theft of, and it affects the Indian farmer, the Indian traders and exporter, and deceives the customers. So this is one example of piracy. So India again challenged that basmati patent, and they had to narrow the claims. So these are real life examples and cases where a lot of confusion has happened, and we had to go and try to protect our rights. Now, when in this scenario, how do you go about developing a technology and commercializing it? You must have a strategy. So you have your inventory of ideas. You'll have to prioritize because everything takes money. So whatever is potential product, you'll take it forward. You'll create a portfolio around one technology. Constantly, you have to create your database and mine the database, see the data, and take forward. Do the analysis. Go for the legal review, business review, technology review, and take forward and commercialize. If you don't want to commercialize, you can out license it. So that's another thing, that you develop a product, you sell it to somebody else, and then let them take it to the market. So that is another way of doing it. So from the invention to invention disclosure to searching for what is prior art, what is existing out there, evaluating the innovation, what you have created, and if it is clear, then you will protect in the form of a patent. Then you will market or you will license it to somebody else, and you will continuously monitor the progress. So there is a constant due diligence required. So if you are an institution, you should have a SWOT analysis. What are your strengths? Then create a culture of innovation, create your think tanks and advisory committees, leverage whatever talents you have, brainstorm the ideas, define what you want to do, and then prioritize which are the areas where you have clear freedom to operate. You have the technologies where you can develop further. And then create your funds. Of course, you need funds to take it further. Create an IP landscape, understand what is there and what is not there. What is not there, you will work on that. So generate your IPs and create a portfolio and go through the whole due diligence and come to a product where you have a product pipeline, and one after the other, you have products coming, and you will then strengthen your brand. So these are the options. This is the logical flow of things that you have to consider and determine whether a particular action will infringe somebody else's IP or not. So that should be the first question, whether anybody else is doing the same thing that I am doing. If not, then you can proceed. If somebody else is doing the same thing, find out what they are not doing and then proceed in that direction. So be well informed, do your state of the art analysis. This will save time, effort, and money from creation to assessment to further on to go till the commercialization. Go to the US Patent Office site, go to EPO, the European Patent site, the WIPO, and search what is out there. Clearly record your documentation is very important. See what is the prior art and never publish. If you want to patent something, never publish. When you go to with the idea to the partners or joint venture partners or to the attorneys, you need to have a non-disclosure agreement. Otherwise, you will contaminate your idea with somebody else and they will use it. So follow your invention disclosure by the provisional patent and then complete specification and so on. And meanwhile, use the time to define your boundaries, what works. But remember, the first inventor to file will get the patent. So please remember that state of the art and prior art, understanding these two things are very important. So any evidence that your invention is already known is a red flag. Does it exist physically? Not required. Even if somebody has described it or shown it in a pilot testing, or they have published it, it is similar to your invention, it's a red flag, you cannot go ahead. Now, ask these questions. Is this invention novel, or are you reinventing the wheel? Is it somebody who already owns it? And who are the players, the different players in the landscape? And it's not important that you have to do everything yourself. You can go for a joint venture, or you can license it to somebody who has all the infrastructure, or you can also do a technology prospecting. Is there a solution that you want to take in and to develop your product? So these things are options that we should know. And it's a complex landscape there. You may have to go and buy some company or a technology. You can see the different options there. Keep in mind these technology life cycles. What is useful, what is not useful. That's what is IP audit. We need to understand what are the business dynamics. Continuously, you have to monitor what's going on, what is happening in the market, what is the feedback in the market, and course correction has to happen. So whatever strategies are there have to be defined by knowledge, and you have to do a continuous technology watch, understand the trends. Is it becoming obsolete? Then you have to start with the next phase and design your strategies. So you create an invention disclosure, you define your novelty, inventive step, utility, you go make a pilot, show the best mode, then you go file your application, understanding what is the state of the art, what is the statement of invention. All these things you have done, then you will come to the pilot testing, develop the product, scale up, test market, and go for commercialization. So these are the steps, and at each step, there is due diligence required. So this is what I wanted to share with you today. Thank you. You can further read this article, which is in this book called Geography and You. It talks about negotiating the GMO minefield. It's written by me. You can further read that. And thank you. Thank you so much. These are my intellectual properties, 5 of my books. Any further questions, I'll be happy to answer. Thank you.
Thank you, ma'am, for that informative talk. Participants, any queries, questions, you can please post in here, or you can unmute yourself and ask. Ma'am, there is a question. Can we patent a new strain isolated from environment, which is not previously known? Mr. Sanjeev asks this.
Okay. So you've isolated something, but what is its functionality? Because when you want to patent something, you'll have to show what is the application of it. So those 3 criteria. What is the utility? So if you have a microorganism, but you don't know for what you're going to use it, just isolation is not enough. You have to show the functionality. So if you know the function of that, then you can go and say that this is a utility, because there are 3 criteria of patentability. You have to satisfy all the 3. So if you isolate, fine. You cannot patent. You show what it is useful for, then you go for patentability analysis further. Okay.
Ma'am, looks like they were the questions. So we will be winding up now. So on behalf of the staff, students and management of Oxford College of Engineering and Department of Biotechnology, I, Salma, take it a privilege to thank Dr. Farah Deba for her valuable time and the information that she's given to us. Hope it has reached all the audience and people do make use of it. Ma'am, on behalf of all of us from this end who are organizing this FDP, on behalf of Dr. Manjunath, the convener of this FDP, I take this privilege to thank you for being part of this FDP session for the second time, because you were there even with Prabhavati ma'am as a discussion panelist. So thank you, ma'am. Thank you so much for being part
Thank
of
you so much. Yeah.
Thank you, ma'am. Dear participants, the portal will be open for 10 minutes for you to fill in the feedback.